That's important. Let's dive into just the syllabus real quick. I just want to sort of just bring us to the syllabus, just to give us a little heads up. So today, 1028, we're working on gene expression. There's an in-class activity that you can do. It's an optional in-class activity. You don't have to do it, but I would recommend doing it. It's kind of a fun activity to do. And there is no homework this week. You get a reprieve from homework till next week, just as an FYI. Now, looking forward, you can see your next exam is coming up on 11/11. Same exact as last time, 40 multiple choice on Blackboard. You'll get 72 hours to do it, which is I felt pretty well on the first exam, so I'm assuming that's probably fine for you guys. And then we after that we have the thing to be heads up about is your rough draft for your BioWiki is going to be due sooner rather than later. So you're going to submit that on Blackboard, and then I'm going to send them out for peer review, and that will be due when you come back from Thanksgiving break. And then your final one will be due near the end of the class. So that's that. Next week, I will tell you, though, that the activity we're going to be doing on 11/4 is pretty involved. So please, before you come to class, read the material that I posted for that week about the genetics of COVID and the epidemiology of COVID, because it's a lot to sort of take in a 45-minute period. So make sure you read it, take some notes on the stuff that you've read, maybe take a look at the questions you're required to answer for the class. That way you know what's going on. Because again, it's a tough activity. It's an important activity, but it is tough. So just make sure you do come prepared with your readings on COVID. Again, they're going to be on Blackboard, just as an FYI. Now, in terms of the lab, you'll notice that you have single transduction and how cells communicate. Make sure you guys do stay up to date on your Labster stuff. They're easy to do, and they do constitute like almost 20% of your grade, so it's a big chunk. You definitely want to make sure you do them. But next week, you'll notice we have a lab that's not on Labster. This one's called amylase. So if we pop on over to our lab materials folder, you go down, 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 and you see week 9 is the activity of amylase. And so there is a pre-lab, /lab quiz that you should do. This will be due at the end of the week, just like all the lab stuff is. There's a lab, there's a procedure, and there's data for you to analyze. So what you're going to do, because this is kind of a complicated experiment to sort of get right off the bat, I did schedule some time next Monday at our normal lab time, so 11/2 at 11 a.m. So I scheduled some time for us to meet on Blackboard, collaborate to discuss this lab, because again, it can be kind of hard to understand until it sort of clicks in your head. So please do come to this if you have any questions. I definitely read it before you come, and then we'll try to answer all the questions we have. We'll walk through what's going on, the point of the exercise, how to do the data analysis, stuff like that, just so everyone's on the same page. And for those of you that can't make it, there'll be a recording of it as well, so you'll have access to that as well. But definitely do try to come on 11/2 to talk about amylase. And just as a note, we'll meet again during the lab time on 11:30, because we'll talk about the next exercise, which is the virtual genetics lab, or the VGL lab, which is again a non-Labster lab. So 11/2 won't be our first meeting as a lab group. We'll talk again on 11:30. Just wanted to bring that to everyone's attention. So definitely try to come next Monday, on Blackboard, collaborate. It won't be that long. I only anticipate it to be maybe 40 minutes. So it's nothing crazy. So, but does anybody have any questions? Whoa. Before we jump in, schedule-wise, grading-wise, anything. All right. So we're gonna do gene expression today. So it's kind of a fun class. How do we regulate the expression of genes? Because you think about you as an individual, right? You eat food and you express genes to break down that food, right? And it's not just you that expresses those genes to break down that food. It's the microbes in your stomach, right? Or in your colon, I should say. And you go to the gym and you work out. You have to express genes. learn biology with me, you express genes, right? You listen to biology, you express genes. You sit on your couch and watch Netflix for 6 hours straight because you're tired. You're still expressing genes, right? When you're sleeping, you're still expressing genes, right? Any action that occurs in your cell, we need to sort of think, how do we control the gene expression? Because we don't want all of our genes to be firing at all the times, right? We have 23,000 genes inside of our genome, each individual one of us. We don't want all those 23,000 genes going and going and going all the time. So we have to find a mechanism to control them, and that's going to be the topic for today. And so we're going to talk, sort of a split lecture. We'll talk about the bacteria and the archaea, how they do it. We use two tried and true examples of gene regulation, and then we'll jump into the wild and wacky world, just basically to talk about how the eukaryotes do it. So, but ultimately, controlling the expressions of genes typically involves controlling transcription, right? And in particular, the initiation step of transcription. So remember, transcription is where we take our DNA template and we use RNA polymerase to make a copy of messenger RNA. Now, typically what happens is we see regulatory proteins, just proteins, they bind to DNA, which either blocks or stimulate transcription. And our prokaryotes and eukaryotes, they do things slightly different, but ultimately prokaryotes are typically doing things to express genes in response to their environment, and you typically are doing things to maintain homeostasis inside your body. And when I say homeostasis, I just mean keeping things balanced, like keeping your internal temperature, your internal concentration of stuff, all that fun stuff in balance. Now, these regulatory proteins, they're proteins that control gene expression, and what they typically do is bind to specific DNA sequences. Now, they gain access to these bases at what we call the major groove, and they're associated they have what are called DNA binding motifs. So we'll go into these 2 points right now. Now, as you should recall from the DNA structure lecture, there's major grooves and minor grooves of our DNA, right? Major grooves where we have much larger gaps between the double helix, and then the minor grooves, which are the much smaller gaps between the double helix. Now, the major grooves, because they're so large, the hydrogen bonds and all the sort of the bits and pieces of the DNA are physically accessible. So, and each of our DNA bases, whether it's A, T, or C, has a specific unique pattern that a protein can recognize. So proteins can read this major groove based upon the space, right? The major groove has a much larger space between it, as you can see here, and then the minor grooves have a much smaller space. So it can read not just whether you have a large groove or a small groove, or I should say a minor groove and a major groove, but it can also physically detect DNA bases present, because remember, each DNA base is slightly different, right? It can detect the C. It knows that the C has to be bound to a G, right? So it can detect what's going on. So these proteins have some level of specificity, some level of knowledge here. Now, this leads us to this idea of a DNA binding motif. And so these are regions of our regulatory proteins that physically bind to DNA sequences. And so the first one we'll discuss is what we call a helix turn helix motif, which is we have two alpha helical segments linked by a non-helical segment. And we'll see that in just a second. And then we have a zinc finger motif, which has several different forms, but they ultimately use zinc atoms to coordinate DNA binding. And then we have a leucine zipper motif, which is a motif that is basically in one subunit of the protein and interacts with a similar region on another subunit. So let's just look at what's going on here. So these are the helix turn helix motif. That's a heck of a thing to say fast as I was doing it. But ultimately what it's doing, we have the alpha helices, and then we have the little turn, like the connector proteins. And the idea being is that these can recognize the grooves based upon it and bind, right? And so these regulatory proteins are things that are binding to DNA after they're being read and can change and influence expression, right? Now we have the zinc, the leucine zipper motif. The idea we have alpha helixes binding, and they have this almost like a zipper-like structure. Where they have, think about I think Velcro is probably the better analogy here, because to me this always looks like Velcro, not really a zipper. but that's just me. See that it binds in 2 spots, it recognizes specific DNA sequences, and then it attaches to another one in that what we call that zipper region. So that's how we recognize our regulatory proteins can recognize DNA. So let's talk about how we can physically use that to regulate our transcriptional processes. So the 2 types of control we can have upon transcription is either positive or negative control, and their name implies exactly what they do, right? Positive control increases the frequency of transcription, and negative control decreases frequency. And what's doing positive control is the binding of what we call activators, which enhances RNA polymerase's capacity to bind to the DNA. And we have repressors, which are involved in negative control, which physically bind to what we'll term as operators, and we'll get into that in just a moment. And they bind to that and decrease frequency or completely stop frequency. So this is what we get. Now, prokaryotes, thinking about what they're doing, they're responding to their environment by changes in gene expression. And it's sort of this interesting thing that prokaryotes do is because of the way their genomes are organized, they typically cluster genes that are associated with this process in a very similar stretch of their DNA. And you might ask yourself, what do you mean by that, Dr. Kearns? Well, what I mean by that is if I have a function, i.e. I'm trying to break down a sugar, potentially lactose, which is the one we're going to talk about, I put all the genes that are associated with the breakdown of lactose right next to each other in a row. And by putting them right next to each other in a row, I can regulate them in real time together. I can express them in real time together. So, which leads us to talking about the Lac Operon. And so for those of you that like dairy products, cheese, milk, pizza, right? Pizza obviously being the best, is all looking at the protein, the sugar found in dairy, which is lactose, right? So some of you might be lactose intolerant, which is a sad thing. But lactose is just a sugar, right? It's like glucose or table sugar, which is sucrose, right? It's a sugar, thus it can be used to get energy from. Now, the capacity for any organism to break down lactose, at least as a microorganism, requires the Lac Operon. And so the Lac Operon itself has 3 genes that we're interested in. We have LAC-Z, which is a beta-galactosidase, LAC-Y, which is what we call a permease, and LAC-A, which is a trans-acetylase. The basic function of these are to import glucose into the cell and break it down into smaller sugars. That's all the Lac Operon really does. You don't need to remember the names, but that's the general function of the Lac Operon. So let's look at this in physical space. Right, we have galactosidase, which is LAC-Z, permease, which is LAC-Y, and acetylase, which is LAC-A. So this is the genes. This is what we would call the coding region. And so these are the 3 genes we're trying to express. We need to express all 3 of these genes in our friend E. coli, which is just a bacteria, to break down lactose. Now, there's a couple other things we have. We have an operator. We have the promoter again. Then we have a repressor protein and something what we call the cap binding site, and we'll talk about that in a moment. And this is all what we call the regulatory system. So the promoter and the repressor and all this stuff, this is what we call the regulatory region. All together, we call these, when we combine the regulatory and the coding region, as the Lac control system. So this is the genetic structure, and this is what we term as an operon, right? All these genes, the ZYA and the regulatory parts all are part of the Lac Operon. And so the reason we talk about this system is, lactose is obviously an important part of human life, but it's a really well-studied system, and it's a really simple system to understand how gene regulation occurs. So it's a really powerful way to understand gene regulation in our microbes. So let's dive into this. So Lac as an operon is regulated negatively. Because it's regulated by what we call repressor protein, right? The repressor itself, remember, just binds to it and inhibits or significantly reduces expression of the Lac-associated genes. Now, the way this works is that in the presence of lactose, there's a inducer molecule which we call allolactose. You can Google what allolactose looks like, but just to know, it's the identical chemical formula as lactose. It just has a slightly different structure. So if you think about, like glucose can have multiple different structures. But what ends up happening is that allolactose can bind to the repressor, and if it binds to the repressor, which again is stopping transcription of the Lac genes, it causes the repressor to come off of the gene and allows transcription to proceed. Now, the interesting thing about the Lac Operon is it's always actually expressed. So even when lactose is not present, the Lac Operon is expressed at a very, very low level. So the repressor is bound to the Lac Operon, but it's still expressing a little bit all the time, which I always found to be really stupid, but it is what it is. Now, in the absence of lactose, we have this really fun system. So we have the lactins here. You can see ZYA. And then we have the Lac repressor, as you can see, is this monster protein, and it's bound to the operator. Now, when it's bound to the operator, RNA polymerase either can't go by, or it takes a long time for RNA polymerase to go by. But the idea being here is that simply the presence of this repressor stops major levels of transcription of the Lac Operon. Right? And the other thing you'll notice is we have what we call a cap, which is about a cap binding site. And now the cap is an interesting protein. Its name is escaping me off the top of my head right now. I'm really sorry about that. But cap is actually as a transcription enhancer, and we'll talk about that in just a moment. So this is what happens when lactose is absent. We have a repressor. It binds, inhibits RNA polymerase from any meaningful levels of Lac expression. Now, when lactose is present, this is where we bring in our friend allolactose. So remember, allolactose, it's an inducer, so it will bind to the repressor. Now, the idea here is that allolactose is essentially a different form of lactose, right? So if allolactose is present, that means lactose itself is also present. So what ends up happening is when lactose is present, allolactose will be present, and it binds to the repressor. Once it binds to the repressor, it causes the repressor to change shape. Right? The repressor is just a protein. And if you cause the protein to change shape by binding to it, it will release from the DNA. And once it releases from the DNA, well, RNA base is not blocked anymore. It will thus transcribe Lac Z, Y, and A, producing 3 proteins, which are the proteins, galactosidase, transacetylase, and permease, which are required to metabolize lactose and thus get energy from lactose. So this is what happens when lactose is present. So we have this dichotomy. Lactose is absent. There's no allolactose present. The Lac repressor is bound. Once lactose is present, we also have allolactose present, which again is an inducer. It binds to the repressor, causing the repressor to fall off, and thus we have the transcription of Lac ZYA. So that's where we are thus far. Now, the Lac story gets a little bit more complicated, but not super duper complicated. Now, what ends up happening when you break down lactose is you break down lactose into simple sugars. You take lactose, which is a relatively complex sugar, and you break it down into things that look like glucose, because glucose can then be used in metabolism, which is a process we'll talk about in a few classes. But just to know, you break down lactose to make something that looks like glucose so that you can get energy from it. Now, when glucose is present, in addition to lactose, E. coli preferentially prefers glucose over lactose. There's reasons behind this, but just to know, if glucose is ever present inside your cells, inside of bacteria cells, you will always use glucose first, because glucose is the easiest sugar to metabolize and get energy from. So what ends up happening, if you have glucose and lactose present, glucose is always used first, and then lactose will be used second. So have this dichotomy, right? Glucose is present, lactose is kind of put to the back burner. But if you use all your glucose and then all you have is lactose left, well, hey, you can now use lactose because there's no glucose present. So that's the idea. It's a preferential choice. Now, what ends up happening is we have this protein, which is the metabolic activator protein, or CAP. It's a protein that binds with a molecule called cyclic AMP, or CAMP. And CAMP is what we call an effector. It's like a think about it, well, this system as like an enhancer. Now, what ends up happening in these cells is when glucose is very, very low or absent from a cell, the levels of CAMP or CP or cyclic AMP increase. And what that does is it causes an increase in CAP, which causes an increase in the expression of lactose, the association of the lactose genes. Now, when glucose is low, when glucose is high, the amount of cyclic AMP or CAMP is lower, thus the organism preferentially expresses the genes to break down glucose over lactose. So it's kind of a weird thing, but let's look at the picture. Let's walk through this together. So in this scenario, we have very low levels of glucose. We have the inducer allolactose present, which means that lactose has to be present, and the promoter itself is activated. So the repressor is not bound to the promoter anymore. And because glucose is low, we have high levels of CAMP. Now, once we have high levels of CAMP, it does bind to that CAP protein, and that CAP protein then binds to the CAP site. Now, the whole purpose of this is CAP is basically like an enhancer. What it does is basically increases the expression levels of the Lac Operon. It also increases the efficiency of the expression. So it basically just makes lactose and the genes associated with the breakdown of lactose go crazy. That's what CAP does. And remember, CAP only binds with high levels of CAMP. And to have high levels of CAMP, you have to have low levels of glucose. Now we can look at the opposite, where we have glucose present. There's almost no cyclic AMP or CAMP, which causes CAP to fall off. It doesn't bind because there's no CAMP because glucose is high. And there's inducer, so there's no allolactose present. Thus, the repressor is still bound, which keeps RNA polymerase from expressing Lac Operon. So we have this really sort of interesting system, right? It's controlled not just by the presence of lactose, it's controlled by the presence of glucose. And the way glucose is doing this is by causing the cell to produce either more cyclic AMP or CAMP or less CAMP or cyclic AMP. So we have this fun system. At least I think it's fun. You guys probably don't think it's fun because you have to learn it, but I think it's pretty cool. It's a really interesting sort of look at gene regulation because the lactose is great and all, but if glucose is present, glucose is way better, way better. So it's sort of like a better energy source. So if glucose is present, you don't want to express lactose. So you have to have some way to turn off the lac expression, and you do that with cyclic AMP or CAMP. So that's the Lac Operon. Pretty straightforward. Remember, that's a version of negative control. But let's talk about our friend, the Trip Operon, which is the genes that are required for the biosynthesis of tryptophan. Now, tryptophan is an essential amino acid. You might recognize tryptophan as like it makes you tired because there's a lot of it in turkey. You've probably heard that before. Not to burst your bubbles, but that's actually not true. There's not enough tryptophan in turkey to make you tired. You get tired on Thanksgiving and Christmas because people eat like pigs. It's not because of the tryptophan at all. So, but I digress. Now, the Trip Operon is not expressed at all when the cell contains sufficient amounts of tryptophan. However, the operon is expressed when tryptophan is low. And the repressor that controls the Trip Operon is one of those helix turn helix proteins that binds to the operator site, which is just upstream of our friend, the Trip Operon. Now, the Trip Operon, like the Lac Operon, is negative regulation. So remember, it's always being expressed, but it can be controlled by a repressor binding to block or inhibit transcription. So let's look at this system. We have more genes involved this time, but it's very, 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 very simple. We have an operator, we have the promoter, we have a gene that's actually a repressor, TRIP. And then we have TRIP E, B, C, B, and A. So we have 5 gene products that are required to make tryptophan in a bacterial cell. And so what ends up happening is we have tryptophan is absent. What ends up happening is the operator is de-repressed, which means that the TRIP repressor is not physically bound. It's not allowing it to bind to the operator, and thus transcription occurs. So when tryptophan is absent, the operon is not bound, and thus transcription can proceed. Now, when tryptophan is present, the operator is repressed. Well, kind of an interesting story here. So we have the trip repressor upstream, and we have RNA polymerase, but we have tryptophan in the cell. What ends up happening is tryptophan will bind to the repressor, causing a change in the conformation or the shape, and it allows it to bind to the operator. But it blocks tryptophan. It blocks RNA polymerase for expressing these genes. So in this case, instead of being the Lac Operon, the repressor was regulated by allolactose. For tryptophan, the repressor is physically regulated by the presence and absence of tryptophan. Right? If tryptophan's present, you don't need to make more tryptophan, right? So you have them bind to your repressor, thus inhibiting transcription. That's, again, similar to the Lac Operon, right? If lactose is present, that means allolactose is present, thus that binds to the repressor. But instead of making the repressor bind, it causes the repressor to come off. So it's kind of an opposite sort of stance from the Lac Operon itself. And so one of the sort of interesting things about tryptophan is it alters the repressor's conformation. And so we have the repressor here. It looks a little bit like this. And it can bind, when tryptophan's not present, but the repressor actually will fit much more snugly when tryptophan is actually present. So when you add tryptophan to the system, when you add it to the repressor, it causes a slight change in the structure, which causes it to fit much more tightly into the grooves of this major groove. Right? So major groove here, the protein fits nice and tight. And just as a reminder, these are our minor grooves. So that's our bacterial expression. We looked at two, they're both negatively controlled, pretty straightforward in terms of like ideas, nothing crazy, but they're they're really important for you to sort of hammer home in your brain and get down pat, because they're something that's not going to go away, just as a matter of the way things are. When you guys take cell biology, one day you'll talk about the Lac Operon again, just much more in depth. So just as a note, know and love the Lac Operon, because you'll be seeing it again and again. It's a model system to understand gene expression and gene regulation. So really important system. So that's our prokaryotes. Straightforward. Now eukaryotes, things get slightly more complex, and so we're not going to dive into all the super duper complex stuff. You'll do that as you sort of get more advanced in your biological learnings. But eukaryotes, as I've sort of foreshadowed already, it's complex, right? And there are some major differences, but there's also a lot of similarities to our prokaryotes as well. Now, eukaryotes have their DNA organized into chromatin, right? So that's, when we think about what a chromosome looks like, right? It can be super duper tightly coiled, and that's what we usually think of when we think of a chromosome. But if a chromosome is not super duper tightly coiled, it's as a form of chromatin, right? And one of the things about chromatin is it's, as you'll see in a moment, it's when DNA is in chromatin, it's wrapped around proteins called histones. And when it's wrapped around proteins like that, it actually complicates DNA-protein interactions. And the other thing that's different about our eukaryotes, different from our prokaryotes, is that eukaryotic transcription occurs in the nucleus, whereas prokaryotes, it just kind of occurs in the cytoplasm. And ultimately, the amount of DNA involved with regulating eukaryotic genes is significantly larger than it is for our prokaryotes. So the big difference between our guys, our euks and our prokes, is transcription factors. And so these are essential and necessary molecules for the assembly of what we would call the transcription apparatus and the recruitment of RNA polymerase to a promoter. And so we have this molecule, it's called TF2D. We'll get into that, what that looks like in a moment. It recognizes a specific sequence called the TA box, or the TATA box. And after I'm just going to call it TF, because I'm tripping myself up trying to say it. I don't know why. But after it binds to the TATA box, a number of other proteins, also all labeled TFs, bind along with it. And then many other transcription-associated factors, or TAFs, bind as well. And then once all that stuff has physically bound, then we can initiate transcription at a minor level, like just a minimal amount of transcription. So just off the bat, you can see this is completely different, right? We're not looking at repressed anymore, right? We're not looking at simple structures, right? There's multiple proteins involved in here. It's not just RNA polymerase coming in and it's doing its thing like in bacteria. There's a lot more to it. Now, transcription factors can be either broad or narrow, and it just varies. So there's some transcription factors that control dozens and dozens of genes. There's others that can only control one gene at a time. It just varies. But specific transcription factors, which only control one or a very small subset of genes, typically, when we think about multicellular organisms, they typically act only in a tissue, like only in your brain, or they only act in a time-dependent manner. And they do this to stimulate high levels of transcription. And well, you can think about what this would mean like in your stomach, right? Say you eat a bunch of food. Well, you need to start the process of transcription to start to break down that food, right? So it's time dependent. You're trying to express the genes to break down that food. You need specific transcription factors to induce the expression of those genes to break down your food. And the basic structure of our transcription factors is that each factor has a DNA binding domain and a separate activating domain that interacts with the transcription apparatus. And let's look what's going on here. So we have an initiation complex here. We have the TA box or the TATA box, and we have RNA polymerase 2, and then we have all these transcription factors, right? All the ones that would, TF, D, B, F, E, H, and so on and so forth. So it's this, quaternary level of protein structure bound to the TATA box itself, or bound to what we would call the promoter. You can see the transcribed region of the gene is up here. RNA polymerase is here, and then this would we consider our general promoter area for our eukaryote. So much more complex, right? Our prokaryotes, just RNA polymerase 2, not all this gobbledygook. Which is why prokaryotes are always the best to start off with. Part of me wishes there was microbiology class so I could just only talk about prokaryotes, but I digress. Now, our promoters and enhancers are sort of two important factors that we need to discuss relative to eukaryotic gene expression. And so a promoter forms the basis of what we call a binding site for our general transcription factors. And these help, as we've discussed, mediating the binding of RNA polymerase to that promoter. Now, enhancers are the binding site of specific transcription factors. And the way this works is DNA bends to form a loop to position the enhancer closer to the promoter. So let's look at what's going on here. So we have an enhancer, we have RNA polymerase, and what we're looking at here again is this enhancer, right? So we have the enhancer and it's bound to an activator. Energy is required here. What it does is it causes a bend or this loop to form. And I love this electron micrograph above because it perfectly illustrates this sort of diagram down below. You can, because you can physically see the loop and you can see the globules of protein and you can see the size here, right? It's really, really tiny. So it causes this loop to form. It causes the RNA polymerase to associate with the activator / enhancer, right? Causing the physical enhanced expression of messenger RNA. Again, it's all just an enhancer and an activator binding, a little bit of energy, start active and efficient transcription of messenger RNA. Next, and then so basically this is what we essentially just talked about, but the idea of the activator binds to the enhancer, the transcription factor binds to the TATA box, and the RNA polymerase binds to the promoter, and we cause that loop to form. So pretty straightforward ideas, but again, more complex than what's happening in our bacterial friends. And so there are some more intricacies to this. So enhancers and activators are all fun and dandy, but we have a new class of proteins we have to talk about, which are co-activators and mediators. And these are also required for the function of transcription factors. So the plot just keeps on thickening for our eukaryotic friends here. So let's go a little bit more into this. So what these do is they bind to our transcription factors, and then they bound to other parts of the transcription apparatus. So it's that multi-protein complex. And then our mediators are essential to some but not all of our transcription factors. And overall, the number of our co-activators and mediators is relatively small, because the reality is you can use both of these things across multiple transcription factors as well. Now, let's talk about that transcription complexes. If we're going to understand these two, we're going to need to talk about that transcription complex. We've talked about it a little bit, but we'll talk about it a little bit more depth now. Now, every eukaryotic gene represents a unique case for this transcription complex, which is kind of a downer, but it is what it is. Now, virtually all genes that are transcribed by RNA polymerase need the same general suite of general factors to assemble at the initiation complex, to assemble an initiation complex. But ultimately, even though it's a similar set of factors, there is, again, part of this is, it's going to be a little bit more specific depending on the gene that you're working at. So gene A is going to have a slightly different set than gene B and gene C, and so on and so forth. So let's look at this molecular region here. So we have the core promoter we can see here. We have RNA polymerase. We have enhancers, the enhancer regions, and then we have the two activators bound. We have an enhancer region here. We have an activator. And you can see we have our co-activator here. It's bound again to our core promoter region. And then you see that transcription complex here. Right? This is all this TAF, right? A, B, F, D, right? That's all our transcription complex. And so, sorry about that. But these, so we have this whole thing. Now you can see our co-activators kind of hop in here. And what these are doing in this scenario are stabilizing this complex by bridging this gap. So they're basically allowing this enhancer to better attach to the transcription complex. Now, remember, these enhancers, once they do bind to enhance the reaction, the synthesis, they will detach. But for them to efficiently attach, they do it better with these co-activators. And then we also have these general activators, E, F, D, and these basically are just part of that general transcription complex as well, and they bind directly to RNA polymerase too. So they're broad, they're general. What's specific are going to be these activators and other parts that aren't sort of the general ones like B, F, and D. But ultimately, the important part to remember here is the big things, right? RNA polymerase is going to do its thing, right? But for RNA polymerase to be turned on, you need activators to bind to enhancers. Those activators and enhancers, those activators, too many words sometimes, they have to bind to this transcription complex and RNA polymerase for it to physically do its thing. And so that's sort of the gist, the sort of the big things to remember. It's again much more complex, right? There's not just a repressor, right? There's this whole complex of enhancers and activators and all that stuff that has to bind before RNA polymerase can do its thing. That's how we regulate things at just the transcriptional level. But there's this, in eukaryotes, this higher level of control, and it ties directly to the eukaryotic chromatin structure. Now, DNA itself is wound around an octet of proteins called histones. When I say an octet, it means 8 of them. And these form when DNA wraps around it. It's what are called nucleosomes. Now, the interesting thing about this, and I thought I had a picture on the next slide, but you can Google what a nucleosome looks like. The reality is, you think about like if you have like a rope, and you tie it around a tree a couple times. That's basically what you're doing to your DNA. You're just wrapping it around some proteins. Now, you can imagine if you have your DNA wrapped around something, like if you have a rope wrapped around a tree, and you need to use that rope, and it's wrapped around a tree, that's going to be really hard to get that rope on the tree, right? It's going to really inhibit your ability to use that rope. And it's the same thing when we have DNA wrapped around histones. When DNA is wrapped around histones and they're forming nucleosomes, it makes transcription harder, because if it's tied up around a histone, you can't express the gene that's attached to a histone. So for you to express a gene that's wrapped around protein, you have to modify the chromatin structure to allow transcription. So again, we talked about like directly regulating RNA polymerase. This sort of level of eukaryotic manipulation, we have to physically get the DNA to unwind around chromatin. I'm sorry, unwind from the histones itself. And so the way we can actually do this, and this is, to be honest with you, this is one of the coolest fields that's been emerging in biology in the recent years. This is this idea of epigenetics. And if you've never heard of epigenetics before, it's a really cool field. So if anything today interests you, I definitely suggest looking it up. We'll talk about it next semester in biology too, because we're going to talk about it in the context of evolution. But in terms of its, where it gets its start, it's all about gene expression and stuff like that. So which is why we talk about it starting now. Now, again, back to the idea of our rope around a tree. If we tie a rope around a tree really, really tightly, it's going to be hard to get that rope off that tree. You can imagine if you tie the rope relatively loose around the tree, you might be able to get that rope off the tree relatively easy. And it's the same thing with DNA. You can wrap your DNA very tightly around those histones, which means it's very hard to get at, or you can wrap it very loosely around those histones, making it much easier to get at. And so this tightening and loosening is what we call epigenetic alterations. And the two types of alterations you can do epigenetically is loosening or tightening the DNA around those histones. The tighter the DNA, the harder it is to get at and transcribe the genes. The looser the DNA, the easier it is to get at the DNA. And the interesting thing about epigenetic alterations is they do persist in the absence of a stimulus, and they can be inherited through cellular division. So this has all sorts of interesting, conundrums for inheritance as well, because you can potentially produce a gamete, right, a sex cell that has an epigenetic modification to it, which can then be passed down. But the cool thing about epigenetic modifications is you can turn them on and off as you need to, but they can also be inherited. They can be temporary, they can be forever, they can be short-term, they can be inherited, they do all sorts of kind of cool things. So let's talk about the most common case we see, which is called DNA methylation. Now, when we say DNA methylation, all we're doing is tacking on a methyl group, so a carbon bound to 3 hydrogens. That's all a methyl group is. We tack that on to our DNA. And what that does is it causes changes in the structure of DNA. And as we've talked about for our eukaryotes, right, it's all about binding of things to the DNA, right? And it's the same thing for prokaryotes, but especially for eukaryotes, right? It's all about proteins recognizing the DNA and binding to the DNA. So if you can imagine, if you stick a bunch of these methyl groups in there, you could potentially prevent a protein complex, the transcription complex, from binding, right? And that's exactly what happens. When you methylate your DNA heavily in a given region or associated with a given gene, it correlates with the inactivity of genes. So more methylation means less transcription of those genes. Less methylation means more transcription of those genes. But more importantly, what methylation does is it causes that DNA to wrap tighter around the histones as well. So it not only does it make transcription go down, but also just makes the DNA harder to access. So being harder to access also drives down transcription. So they are related as well. So that's pretty cool, right? You can methylate your DNA and change expression, right? You can regulate your genes just by adding some simple chemical signatures to them, which I think is pretty fascinating. Now, there are other epigenetic modifications as well outside of DNA methylation. They include acetylation, they include ubiquination, and they also include phosphorylation. We're not going to talk about those just as a note, but when you take cell biology with me, maybe next year or the year after, you'll definitely talk about those. But stay tuned. It's a never-evolving story with your biology degree at Fisher, I guess. And the other thing we can actually see for our chromosomes is what we call X chromosome inactivation. And this occurs in mammalian female, so those that are XX. Right? What they do, and this is true for humans as well, not just like cats and stuff. What ends up happening is they inactivate one chromosome. So they have two X chromosomes, they inactivate one of them. And the reason they do this is it's, as the the text says, it's a dosage compensation. And what that means is, males, at least for humans, we have XY, right? So we only have one X chromosome. And our Y chromosome is pretty useless. There's only genes required on your Y chromosome as a male for the development of testicles. That's really it. That's pretty much all the Y chromosome has. It's pretty unimpressive. So males only have one X chromosome, so they have all the genes they need on that X chromosome. Now, having two copies of the chromosome means you have double the genetic material that's found on that X chromosome, which means you potentially have double the amount of protein produced. So to limit the amount of protein that you can produce from what's on your X chromosome, females inactivate one of them. And the way this works is there's a non-coding RNA called XIST or XIST. It coats the entire inactive X chromosome, leading to an overall histone modification, and thus a complete shutting off of that X chromosome itself. So kind of a fun thing. I'll also make note about those of you that have like female cats. X chromosome inactivation is what gives some cats certain colors. Like, so for instance, calico cats, they have their colors the way they are because of X chromosome inactivation. So those of you that like cats, now you know why. Now, so that's the basics of histone modification and epigenetic modification. As I mentioned, there's a lot more to it, right? So histones are, are 4 subunits together. So 8 total proteins, but 4 smaller subunits. And so for any organism that wants to modify their histones, there's 4 possible histones that can be modified. And as I mentioned, you can acetylate them, you can methylate them, phosphorylate them, you can also ubiquinate them. So there are other modifications that we'll learn about later in your biology careers. And there are all sorts of interactions with these histone modifications and potential co-activators and activators and all that fun stuff. So there's a lot more to this story of epigenetic modification, but I want to just sort of show you what this looks like. So we have our histones, and you can see they're just globules of protein bound together, and your DNA wraps around them, right? Again, like the rope around the tree example that I mentioned. And that gives DNA some rigidity and some overall structure. But what you can do is tack on to those histones. In this case, it's in a C. This could be a methyl group. This could be a ubiquinol group. It could be any number of things, which causes essentially the DNA to essentially become split further apart. Right? You can see that the difference here, right, between this and this, right? This has acetyl groups. This doesn't, right? So causes a really radical change in how tightly packed those histones are relative to one another. So this is sort of how we get at this idea of epigenetic modifications. It just allows things to become more accessible. And there are, more sort of things we should discuss relative to chromatin remodeling itself. So we have what are called chromatin remodeling complexes. And these are large complexes of proteins, and they modify histones in DNA, and they change chromatin structure. So these are ATP dependent, means they require energy, and they function as what we call molecular motors. So when we talk about it's in one of the later classes we're going to talk about motor proteins. But it's pretty cool. It's a pretty cool topic because motor proteins move things around the cell, and so we'll talk about this again. But that's what these chromatin remodeling complexes do. They're molecular motors. And what they do is they catalyze 4 different changes in DNA histone binding and make DNA more accessible to those regulatory proteins. And so what they do is a couple different things. So it requires energy. So we have this basic level of a histone. So it does 4 different things. It causes, the easiest one to understand is histone displacement, where it causes the histone to detach. You can have histone replacement, which is where you substitute one histone for another. You can cause nucleosome sliding, which is when you move the entire structure up or down, which again would change how the DNA could potentially be accessed. Or you could potentially remodel it, causing it to, cause it to be, tighter wound or less tighter wound. Again, it's all based upon these remodeling factors. I like watching. There's cool videos out there if you have like, a couple minutes to watch them about nucleosome sliding. It's a really cool video to watch because it actually is like a zipper moving up and down a piece of DNA. It's actually pretty cool. But that's that. Now, what we talked about thus far has all been regulation of transcription, right? Whether it's the Lac Operon, the Trip Operon, regulators, right? That was all to regulate transcription. Now, the other aspect of eukaryotic regulation is what happens after we transcribe messenger RNA. So how do we physically regulate the after effects of messenger RNA, right? So what happens when we make a bunch of mRNA? How can we control gene expression after we make mRNA? So we do this with a couple of different things. So we have small RNAs, which are classified as miRNAs and si or siRNAs. We're not going to talk about those today. They're a pretty in-depth process. Just as a note, they're relatively new in terms of a topic. So if you want to read more about this, I linked this article from NCBI, which is the National Center for Bioinformatics and Information. It's the American governmental database for science, literature, and all that fun stuff. So if you want to read more about mi and siRNAs, take a peek here. But the ones we're going to focus on are alternative splicing, RNA editing, and mRNA degradation. Now, as you should recall from a previous class, when we talk about the structure of eukaryotic genes, you should remember that eukaryotic genes have both exons and introns. So exons are the part of the DNA that actually are made into mature messenger RNA that will then be translated, right? So those are the things that are actually used to make the proteins. Now, introns are what we call intervening sequences, and these are spliced out. So they're removed. So you can see in both these examples, we have introns are in purple, exons are in yellow. There's the 5' cap we've discussed, and the 3' poly A tail. Remember, these are stabilization and protector artifacts. And you'll notice is that we have exons are only present in the mature messenger RNA, which is then used to make a protein. Now, alternative splicing is a cool phenomenon where you choose specific types of exons to make your protein. So you can see we have exon 1, 2, 3, 4, 5, and 6 in this primary messenger RNA transcript. But you can see in our mature mRNA over here, which is in your thyroid, we have exon 1, 2, 3, 4. And in your mature mRNA in your hypothalamus, we have exon 1, 2, 3, 5, and 6. So this is what we call alternative splicing. We take the same messenger RNA, we cut out exons slightly differently, and we get two potentially different genes. So in this case, we have calcitonin in your thyroid, and in your hypothalamus, we have CGRP, which is the calcitonin gene-related peptide. So two genes, so the same gene, transcribed the exact same way. The exons are spliced in a slightly different way, giving us two distinct proteins that have two distinct functions. And those distinct functions are related to where they are, in your thyroid or in your hypothalamus. So, two parts of your body that are really important for regulation, right? Your thyroid is really important for regulation of hormones and stuff like that, and your hypothalamus is important for the regulation of, well, almost everything. It touches everything. Hypothalamus is kind of a jack of all trades. So that's alternative splicing. It's pretty straightforward, right? Just cut out different exons and you get different protein products, right? Because remember, the structure of the protein is directly tied to the sequence of the mRNA. If you change that sequence of mRNA, right? In this case, we have 5 and 6. We have 4 over here, right? That addition of more amino acids is going to change the structure of that protein. So very, very simple, but as you can see, it's a very powerful process. And it's one of the really fascinating things about eukaryotic genes is if you look at the human genome, right, we only have about 23,000 genes or so. but we know that humans make almost 100,000 proteins. And the way we make 100,000 proteins is via alternative splicing. Every gene can be spliced in at least 2 different ways, some multiple different ways. So every gene can be alternatively spliced, which gives rise to a lot of cool proteins, which I think is, I think it's amazing, but again, you don't have to think it's it. That's that's alternative splicing. Next up, we have RNA editing. And so what RNA editing, at least in mammalian individuals, involves what we call a chemical modification of a base to change its base pairing properties. And so what does it mean? Well, we're changing a specific base. So for instance, in the mRNA, you have for serotonin, which is 5-HT receptor. So it's the brain receptor for opiates. It's edited at multiple sites to produce 12 different forms of the protein. So you're changing, say, a T to a G, or a U to a G, or an A to a U, right? You're just changing some bases, and by changing some bases, you create slightly different changes in amino acids, thus slightly different proteins. So this is what mammals do. We can slightly edit our mRNA, producing slight variations of proteins. So slightly, what we call isoforms, are slight modifications in the structure. Other types of post-transcriptional control, it's an interesting thing. Gene expression can be regulated up to the point where it crosses the nuclear membrane. And then in terms of the initiation of translation, you can sort of also modify that. So when translation occurs, and so for instance, if we have a mRNA called ferritin, it's involved, as its name implies, ferra meaning iron. So it's a protein that's involved in the acquisition and usage of iron. And so this ferritin mRNA only translated, so it will only go to the ribosome if iron is present. And when I say iron is present, I mean iron is present inside the cell. And the other thing that we also see is that mature messenger RNAs, so the ones that have their introns spliced out, have various half-lives depending on the gene location and the gene and the location of expression. So messenger RNA products last longer in certain parts of your body than they do in others, and some genes last longer as messenger RNA than some others do. And what ends up happening is when something degrades, it starts with the degradation of the poly A tail, so that 3' end. And once you lose the tail, it leads to the destabilization of the messenger RNA, and thus the destruction of mRNA. So those are the other ways we can modify our messenger RNAs, but we can also directly modify and degrade our protein. So pretend we produce a bunch of protein, and we don't need it anymore. Because remember, if a protein is hanging around, it's going to keep doing all the functions that protein was doing. So proteins are continuously produced and broke down and produced and broke down. They don't just hang around indefinitely. It's very much like messenger RNA, right? They don't hang around forever. And so what ends up happening is we have a class of molecules called ubiquitin. And what they do is they require energy, so in the form of ATP, to bind to proteins. And once you have ubiquitin, this process of adding ubiquitin to proteins is called ubiquination, as you can imagine. Once you have enough ubiquinin bound to a protein, it's then destroyed by a process called proteolysis or proteolysis, whichever you want to call it, which is just the destruction of protein. And these happen at lysosomes, which you think of as like a slight variation in a lysosome. So it's one of those micro bodies, like a lysosome or a perioxisome. And we can actually physically look at that. So we can look at degradation by the ubiquitin proteasome pathway. So we have a protein of interest. Energy is required to stack on ubiquination. That goes into the proteasome. It's shredded into small fragments. To get the ubiquinins off, it requires some energy. To break them apart requires more energy. And then we require for this process to start all over again. We have an enzyme called ubiquinone ligase, which tacks on things, ubiquinons onto targeted proteins for degradation. So we can regulate both the destruction of messenger RNA. I will make mention, though, that the enzymes that do break down RNA are called Rnases, RNAASE, Rnases. As their name implies, they break down RNA. So we can have multiple sort of ways to regulate post-transcriptionally what happens inside of a eukaryotic cell. Now, these processes do occur in bacteria as well. So ubiquination does occur in bacteria. There's no secondary splicing in bacteria, right, because they don't have introns. There's not mRNA editing in bacteria. So bacteria have a limited number of ways to regulate their expression and their translation after the fact. But they do it all before the fact. Because the reality is, remember, a bacteria and an archaea is subject to its environment. You as an individual, as a eukaryote, is something that's larger. You're mostly just trying to maintain your internal self, right? Your internal temperature, your internal solute concentration, and so on and so forth. So this is a sort of a nice little summary slide of our gene expression and control mechanisms in our eukaryotes. So we have transcription itself. It starts in the nucleus. Once transcription occurs, and we discuss all the mechanisms by which transcription can be regulated with our enhancers and all that fun stuff. We have RNA splicing, and remember, we can have alternative splicing to cut out different exons and use different exons. But ultimately, we produce that mature messenger RNA. Again, remember, every mRNA has multiple alternate forms of mature mRNA. Now, that mRNA can be degraded by enzymatically if you want to regulate that itself. Sometimes, remember, as we discussed with the presence of iron, some mRNAs can only be expressed or translated if that specific compound is then present. But once it leaves the nucleus, it heads to the ribosome, and it's synthesized into a protein. And once it's made into a protein, it is subject to ubiquination or destruction by ubiquinins and the proteasome complex. So a number of different ways that we can regulate gene expression. Again, much more complex in the eukaryotes. We went over the Lac Operon and the Trip Operon. Those are pretty straightforward. But again, much more complex in our eukaryotic friend relative to our prokaryotic friends. So that is going to conclude, I guess early today, of our discussion about gene regulation as a process. I know that's always exciting when we end early. So as a reminder, let me actually share my screen one more time. So there is an in-class activity for you to take a peek at. So if you go over to weekly modules, gene expression, there is a weekly activity here. It's called Alien DNA. It is an optional activity for you to do. You don't have to hang around to do it. You can do it on your own. I recommend you do it because it will really reinforce your knowledge about DNA binding and the order of DNA and all that stuff. So again, not required. I definitely recommend that you do it, you answer these questions and post them to Blackboard. You do it on your own or you do it in a group. That's up to you. But I definitely recommend that you do it. But again, it's not required for you to do. But I think it's kind of a fun. It's kind of a fun thing because it's all about exobiology, and I find that particularly very fascinating. Other than that, I will hang around for a bit if you guys want to chat. I can also start the breakout rooms if you guys want to do this as a group as well. Like you want to go break out with some of your friends or something like that. Again, you can do it on your own time if you don't want to hang around. But yeah, so like I said, you can hang around and do the assignment. You can do the assignment on your own, or you don't have to do the assignment at all. It's up to you. So otherwise, I'll see you guys next Monday. Remember, we're going to have a meet and greet for our amylase lab, discuss it, and all that fun stuff. You guys have a great weekend. And I should stop recording.